So today, the question we have to address is whether there is a personalized approach of COVID-19 and whether we could predict who are the patients at high risk of severe COVID-19. We know that they are mostly males, aging patients with often comorbidities, different comorbidities. There could be a genetic predisposition. There could be also predisposing autoantibodies against interferons. So what else? So this is what we are going to discuss. And I will call to the floor the first speaker, who is Jacques Fellay. A physician scientist in Lausanne University Hospital. He is an expert in the role of human genetic variation in the response to pathogens and the expression of infectious disease. He is leading the Precision Medicine Unit of the Lausanne University Hospital, and he is the co-director of the Health2030 Genome Center in Geneva. He is also a member of the Swiss National COVID-19 Science Task Force, and he has been part of the consortium that describes the role of defective interferon pathway in severe COVID-19. Please, Jacques, you have the floor.
Well, thank you very much. It's a pleasure to be here and to open this round of presentation. Thank you, Eric, for the introduction. Let me tell you about the ground layer, the basics of what is needed to understand when we want to talk about precision medicine approaches to any disease, actually, and to COVID-19 in particular. We need to understand differences in germline DNA that could have an impact in interindividual differences. So host genetics of COVID-19 is my topic. First caveat. We are not covering everything when we talk about host genetics, obviously. When we talk about susceptibility or severity of an infection, we have this confluence between everything on the pathogen side. Let's remember that infectious diseases are the paradigmatic environmental diseases. You need exposure, otherwise there is no disease. But still, there are still also genetic disease, which makes them fascinating. So virulence of the pathogen, not that much difference as far as we know on the SARS-CoV-2 side, and except with some of the new variants that are question marks on that side. And on the human side, on the host side, there are a bunch of non-genetic factors, most notably age, obviously, comorbidities, and there are some genetic factors that we want to understand for a bunch of different reasons that can be grouped in two main categories. When we interrogate the human genome for biomedical research, we have two goals in mind. The first one being to better understand what's going on at the biological level, understanding biological mechanism, because we put our fingers on a specific place of the genome that varies, helps us develop new diagnostic therapeutic approaches and a better understanding of the disease. And obviously, we also hope to be able to develop individualized prediction models. That's the most hyped and most promising now precision medicine approach, where we can be better at predictive and preventative medicine. So why is COVID-19 a good stage for host genetics? The most obvious answer is that it's a very variable disease. So this figure was just done by Sophie Limou for the COVID-19 Human Genetics Initiative. And it shows that really in the population, you have asymptomatics, you have post-symptomatics, you have severe, you have fatal disease. And this is true even if you get rid of the known risk factors. So even if you look at, let's say, less than 50 years old people without any comorbidities, you'll have the whole spectrum of potential outcomes. This means that it's a good phenotype, it's a good outcome that we can interrogate in terms of potential impact of human genetic variation. And an early argument in favor of that was published as preprint already in April last year, later in the paper shown here, done by Tim Spector and the other people working on UK twin studies, where they showed that the self-reported symptoms of COVID-19 are actually heritable. That's a strong indication that what we observe as differences in the population could be caused by shared ancestry, shared genetic factors in the human genome. So what has been done? And The amazing things about human genomics, since decades actually, but it is even more true now, is the international collaboration. Open, friendly, I have to say. And there has been a massive international mobilization since day one of the pandemic. I cite two large-scale initiatives I'm lucky to be part of. There are hundreds of scientists in each one. The COVID-19 Host Genetic Initiative, which is coordinated by Andrea Gana and Mark Daly in Finland. And the goal is really to understand genetic determinants of COVID-19 susceptibility and severity, and also to provide this platform where everybody can share in real time their data and genetic results. And the second one is coordinated by Jean-Laurent Casanova at Rockefeller and Helen Su at NIH. And this is called Human Genetic Effort. And here the goal was more focused on the extreme cases. So those people that were really sick that when they shouldn't be. So young people with intensive care unit admission or death, for example, trying to find rare genetic variants, most often Mendelian, single gene genetic defects that would explain the severity of the disease. So let's go for a very short overview of what has been found. So the first finding came from China, actually, also in April or May last year, when a group looked at ABO locus specifically without using genome-wide strategy at the time, and found that the risk of SARS-CoV-2 infection was increased if you have a blood group A and decreased if you have a blood group O. So the effect size is small, so it doesn't really matter for an individual, but at the population level, it was there. And this has been discussed for a while and then confirmed, validated in several genome-wide association studies later on. And here, the Manhattan plot I'm showing with the human chromosomes on the x-axis and the strength of the genetic association on the y-axis, you can see there are two regions, including ABO locus on chromosome 9. And the second region that was found to be associated in this first G was the chromosome 3 locus, encompassing several innate immune genes that is still today the strongest genetic factor at the population level. So for ABO, there is no association with the severity of the disease. It's just acquiring versus not acquiring the disease in the first place. And the mechanism itself is still unclear. ABO is important for several other viral infections. We already know it. And there could be some transequitial effect. So some polymorphism, variation in the genome at that place that control the expression, the amount of proteins that are involved in innate immunity elsewhere in the genome. So then the common theme of most papers that were published since then, there are like 200 papers now published on human genetics of COVID-19. It's the findings in viral entry, immune responses, and inflammation. Those are the broad categories that have been identified as important in the human genome. And this is a figure from the genomics study paper published in Nature late last year, where very severe cases were selected and compared to controls. And here, for the first time, there were several places across the genome that were significantly associated with severity of COVID-19 infection, including this chromosome 3 locus, where you see a bunch of gene names here, CCR2, CCR3. As soon as you see CCR or CXCR, something in the genome, that's usually innate immunity. And you have also interesting things like TYK2, which was already known to be associated with some infectious diseases, and interferon receptor gene. The interferon word you'll hear about a lot. You already heard from Eric. Interferon is part of our first line of defense against viral infection. It's a little bit when the alarm is rung in the cells, in the organism, interferon will be released and tell the immune system to be awake and to fight. The Human Genetic Initiative has now compared a large number of cases versus immense numbers of controls. And here is one of the analyses released in November. Now release 5 was just made, but with less beautiful figures. That's why I didn't include it yet. It's about 10,000 cases versus more than 1.5 million controls. And that is possible thanks to the participation of industry as well. Like those large companies having profiles from customers that participate in those questionnaires and are part of those studies now. And it was a confirmation of the genomic hits and additional places in the genome that are important. Again, inflammation, immune response. And some of them were absolutely new in terms of viral diseases. They were known to play a role or to be potentially active in immunity, but never for viral diseases before. The other consortium looking at those exceptional patients that shouldn't be in the ICU when because of their age and absence of cofactors was able to identify rare errors of immunity, basically holes in the immune system that made people very susceptible to that infection at that moment in their life. And 3.5% of patients had rare variants in genes from TLR3, IRF7 pathway, leading to an impaired interferon response. And this gives an idea of potential therapeutic option, which would be in that case to substitute the lack of interferon in those patients. So the clinical trials done with interferon are not very convincing so far, but interferon was given late. So here it would probably need to be given early in the disease to have an impact. And the mirror image of that was the second paper published by the same consortium, where autoantibodies against interferon were found in 10% of those rare patients that had severe diseases. So it's a different biological mechanism leading to the same outcome, not enough interferon to get rid of the virus when you need to get rid of it. So dysregulation of the immune system as a consequence. So as a conclusion of this host genetic part of what has been done so far in COVID-19, human genomic diversity is contributing to the striking individual differences observed in COVID-19 susceptibility and severity. It is a very important tool to uncover molecular mechanism all over human biomedicine, but also for COVID-19. But I want to insist now that genetic diversity is by no means an excuse for the extreme socioeconomic differences we observe in disease risk everywhere on the planet. So genetic is useful as a tool to understand biology. It's not an explanation as to why there are differences between populations on Earth between humans. And that's all I had for this initial presentation. Thank you very much.